Okay, folks, let's talk about basic concept, exocytosis, endocytosis. Um, the big thing to remember here is that cyto means cell. So we're talking about what's going on for the cell. And exo should remind you of exit, where you're leaving. So exocytosis is making stuff leave the cell. The cell do this to get rid of waste. It might do it to release chemicals that it makes. Think of neurotransmitters or hormones, things like that. If we're going to give them off, get rid of them, exocytosis would be how to do it. And this picture over here, this is exocytosis, where you see we have a chemical, maybe a neurotransmitter that we've made. We collect it into a vesicle. We send that vesicle up to the plasma membrane. And when it touches the plasma membrane, it breaks open and spills. I should do that further away there. It breaks open and spills, and all of the contents are released to the outside of the cell. So that's exocytosis. Now, the opposite of exocytosis would be endocytosis, where you bring things into the cell. Endo means bringing in, cyto means cell. And any of the ways that we're bringing things into the cell will be endocytosis. But we tend to go with subcategories. We name for subcategories. So here in the middle on this diagram, we have pinocytosis. If you're taking in taking in fluid or small particles in fluid, we say pino. So pinocytosis, because pino means drink, and we still use that word for non-biology things too. Think of famous wines like Pinot Noir or a Pinot Grigio. Those are Pinot, to drink. Okay. Now, large particles. Think a white blood cell grabbing and taking in a whole bacteria, like you see going on, that type of thing here. We use the word phago. Phago, to eat. If a cell is taking in a big thing, that is it's doing phagocytosis, and the little membrane-bound area holding what they took in is a phagosome because it's doing phagocytosis. Okay, so that's pretty straightforward. That makes sense. But here's the third one. It's a little weird. If we think of phagocytosis as being to eat, we have one for the picky eater. We name it differently. This is receptor-mediated endocytosis. This is endocytosis, but it won't take something in just because there's something there. It'll take something in if and only if the thing matches the receptor that is on the surface of the cell. So let's take a look. Here we have receptors on the surface of a cell, all right? The little green things right there. And if the blue, whatever these chemicals are, touches the receptors, they don't care. Those don't match. But if the little star-shaped ones touch, there's the receptor grabs on and holds it like, oh, this is what I need. And at that point, the cell says, oh, let's pull that in. And it creates a vesicle that has the substance it's been looking for, that the receptors say they want it. So receptor-mediated endocytosis, it's still endocytosis. You're still taking something into the cell, but it has to be something that the cell is looking for and has receptors on the surface of the plasma membrane. Anyway, I hope that makes sense. Exo is getting things out of the cell. Endo is taking things into the cell, and fluids would be pinocytosis, type of endocytosis. Large particles would be phagocytosis. That's also a part of endocytosis. And receptor mediated, that's the third type of endocytosis. I hope this cleared that up for y'all, and we will talk again soon.